Igor Yakomenko is currently Professor of Environmental Science at the National University of Food Technologies in the Ukraine. He's published a very important paper on oxidative effects of low-intensity radiofrequency radiation on living cells.
Hello, everyone. Glad to be here. First of all, thank you to all organizers for such a great event. I think it's really important for medical, for scientific communities. And I am here to present you some ideas about oxidative mechanisms of health effects of low-intensity radiofrequency radiation. First of all, let's discuss a little bit the main idea. What is oxidative stress? What is reactive oxidant species, antioxidant systems, and possible oxidative effects of radiofrequency, low-intensity radiofrequency radiation? I'm pretty sure that most of you know that oxidative stress is actually imbalance between reactive oxidant species production or overproduction and the ability of cellular antioxidant system to detoxify these reactive oxidant species. Look at these apples, a great illustration, yeah? From fresh, nice apple to this really kind of rotten, it just may be due to too much oxidation in these living cells, and actually we may have such situation in our own living cells because oxygen is always in our cells, you know, and sometimes something may go wrong and we will have these conditions. Let's discuss what is actually a reactive oxidant species or ROS, which may result in oxidative stress. It's a group of molecules, mostly free radicals, which always contain oxygen and which are very chemically active. For example, superoxide radical or hydroxyl radical or other forms. For example, hydrogen peroxide isn't free radical but reactive oxidant species. But in most cases, people use interchangeably the ROS and free radicals ideas, just because most free radicals in our cells, which are critically important, are ROS and opposite. So again, a lot of ROS we can get just through water molecules, just through, for example, some effects on water molecules. Let's say ionizing radiation can break water molecules, and we have a lot of different forms of free radicals, which are actually very, very aggressive in our cell environment. And about antioxidants. Antioxidant system actually consists of two main parts, enzymatic and non-enzymatic parts. Again, it's similar to most of us, I'm pretty sure, that enzymes, antioxidants like superoxide dismutase or catalase or glutathione peroxidase, they are enzymes which protects us from these aggressive forms of reactive oxidant species, transforming them in most chemically, let's say, stable, not so chemically aggressive. Non-enzymatic antioxidants, it's mostly vitamins, for example, vitamin A, vitamin C, vitamin E. The most powerful antioxidant is vitamin E. Some minerals like selenium, some phytochemicals, and even coenzymes, for example, coenzyme Q10. Again, the main idea for normal situation in our cells, we need a balance between ROS production and activity of antioxidants, both enzymes and non-enzymes. And when we have this imbalance, it's the situation of oxidative stress. Here, just a couple of ideas. The main enzymes, antioxidants, for example, superoxide dismutase, it particularly defends us, protects us from superoxide radical, transforming it in less aggressive hydrogen peroxide. And then we have other enzymes, catalase and glutathione peroxidase, which protects us from hydrogen peroxides, transforming it actually in water. And again, in presence of ferrum, we have some risks of hydroxyl radical production. So again, today we know a lot about this chemical situation and ROS production and control of enzymes on this situation. General picture. Which factors, which situations may stimulate ROS production in our cells? A lot of environmental factors, starting from ionizing radiation, air pollution, smoking, UV. And interesting thing that, for example, our own immune system, let's say white blood cells, use ROS to protect us from viruses and bacteria. We have a special enzyme, NADH oxidase, which actually produce superoxide to kill bacteria. But during long-term inflammation, long-term infection, this ROS may be harmful for our own cells. That is why it's a challenge. And again, metabolic most maybe understandable metabolic way to ROS production is in our mitochondria, in our power plants, because we always have oxygen there. In normal situation, we have transformation of oxygen into water just due to oxidation of organic molecules and getting ATP. But sometimes we have not water, but just O2 with one electron, superoxide, and that is why it's actually the beginning of this ROS production. And if we have some environmental factors which may affect both, for example, inflammation situation or this metabolism change in mitochondria, we definitely will have a risk of ROS overproduction. Okay. And here some simple scheme on connection of oxidative stress in our cells with health conditions, with diseases. And today, actually, in scientific literature, we may find about 200 diseases which are connected with oxidative stress, starting from skin irritation, burning, dermatitis, and finishing, for example, Parkinson, Alzheimer, and cancer or aging. So a very broad background. That is why it's really important to understand that if we have condition of oxidative stress in our cells, we have a real risk for many diseases, at least in long-term situation, to be developed. And here, for example, again, one simple way to get ROS overproduction in our cells is just from water molecules through the radiolysis with ionizing radiation. It's very important to understand that radio frequency, microwave radiation, is non-ionizing. It doesn't have enough energy to directly break water molecules. But yet in 1994, some guys demonstrated experimentally that low intensity microwave in some, let's say, strange condition may actually produce, again, ions from water molecules in so-called friction between clusters of water molecules. The idea was that clusters of molecules, due to increased movement in some tiny percentage of microwave energy, may actually result in this breaking of water molecules, and it was demonstrated experimentally in fresh water and in water solution that it's actually possible. It's one simple, let's say, physical possibility to have ROS in our cell just from water molecules, because we have a lot of water molecules inside. Next, it was a more kind of biological, biochemical approach. In 2007, Israeli researchers demonstrated on the model of HeLa cells that actually NADH oxidase may be activated under low intensity radiofrequency radiation. NADH oxidase is exactly that enzyme which produces superoxide radical, you remember, in white blood cells. Yeah, but actually we could find these enzymes in many different cells in our organism, and it was kind of one of the first clear demonstration that low intensity radiofrequency radiation actually may result in overproduction of ROS in our cells. Next, a few years later, it was demonstrated that mitochondrial pathway as well may be involved in ROS overproduction under low intensity radiofrequency exposure. It's Australian researchers demonstrated on the model of human spermatozoa that actually ROS overproduction in mitochondria started much below the so-called safety limits of radiofrequency radiation, and it again was clear that we have a few different possibilities to ROS overproduction under low intensity, so-called non-thermal intensity of radiofrequency radiation. During last decade, a lot of research in different laboratories over the world actually were dedicated to analyze the possibility of oxidative stress development under low intensity radiofrequency microwave exposure, and general and typical indices is ROS overproduction, including some particular ROS, level of lipid peroxidation, the most may be important oxidative damage of DNA, and activities of antioxidant enzymes. Later I will give some statistics, but just now I want to demonstrate just a couple of simple metabolic examples why ROS may be really harmful for our metabolism. Maybe one of the first ideas was developed, it's about lipid peroxidation. Interesting thing that one single molecule of hydroxyl radical may result in hundreds of lipid peroxides from unsaturated lipid. What is unsaturated lipid? It's actually a part of our cell membranes. Yeah, phospholipids is actually the main structural element of our cell membranes. And if we have in this situation, instead of normal lipids, we have this lipid peroxidase, it's actually a big problem because it may affect barrier function of our cell membranes, and you know it's a lot of risks for cell metabolism. It was one of the first understandable idea how ROS may affect all our cellular metabolism. And maybe the most important issue is about DNA damage. Back to, for example, ionizing radiation, we definitely know that we have two main mechanisms of ionizing radiation DNA damage, so-called direct and indirect damage of DNA. Direct when ionizing radiation directly affect DNA, and indirect through the water molecules. Under ionizing radiation, water molecules may be broken, produce reactive oxygen species, especially hydroxyl radical. And here we will get this oxidative damage of DNA in indirect way. And interesting thing that this indirect way much more often, much more possible than direct, just because we have a lot of water molecules in our cells, and because this hydroxyl radical is so chemically aggressive. And now it's an important issue. It's about ionizing radiation. But if we have possibility to get this hydroxyl radical from non-ionizing radiation, including radio waves, from other environmental factors, we will get exactly the same situation as under ionizing radiation exposure. I think it's totally understandable and it's very, very plausible idea. And now how actually hydroxyl radical may affect our DNA. Again, it's demonstrated very clear mechanism. For example, one of the generally accepted idea that hydroxyl radical may affect guanine, one of the nucleotides, transforming it in oxaguanine. Again, it seems like not a big deal, just instead of H, we have O here, but due to this additional oxygen atom, this oxaguanine now may be wrongly complementary not to cytosine as normally, yeah? But also due to opposite side of these molecules, it may be complementary to adenine. And it's total disaster for DNA transcription, for DNA translation. Why? Because during the replication, actually, we always need to get against C, G, against A, T. If, for example, under Ross attack, we have oxaguanine, just next replication, we may have here in this point of dotted DNA, A instead of C, and next AT, just totally different pair of nucleotides, and we may have a lot of such points on the DNA, on the dotted DNAs, and we are talking about so-called genome instability. And it's the direct way to a possible cell carcinogenic transformation. Yes, we have some repairing mechanisms, we have enzymes in our cells, which actually protects us from this situation. And sometimes it works, sometimes it doesn't work. For example, in some cases, improper repairing actually may result in removing these nucleotides, and actually we will have single strand breaks in DNA molecules. And if we have single strand breaks, it's much easier to get double strand breaks. Actually, we have physical damage of DNA molecules, physical damage of our chromosome. And here, by the way, the demonstration from our laboratory, it's a model of quail embryo cells, very sensitive, very simple model, biological model, and interesting thing that just due to few days of commercial cell phone radiation exposure, we got this significant damage of DNA. It's so-called comet assay. Actually, it's a native nucleus of cells, and here so-called tails of comets, we have actually these broken pieces of DNA, and definitely we see that the damage was significant. And again, I repeat, it was just after normal, let's say, commercial cell phone radiation exposure. So actually here we have this situation with double strand breaks of DNA. Okay, and again, a couple of examples from our experiments on this model of quail embryo cells, on quail embryos in ova. We demonstrated, for example, that again, just due maybe 2 weeks of commercial cell phone radiation exposure, we have significant 2-3 times increased level of superoxide overproduction. And we think it's just about mitochondrial pathway, because our way of detection was close to mitochondria metabolism. And again, we demonstrated that this oxaguanine, which is the marker of oxidative damage of DNA, was significantly increased in this quail embryo cells after low intensity radio frequency radiation exposure. So we actually confirmed a lot of these ideas and data from other laboratories. And actually today a lot of discussions, and especially in media, especially in some kind of non-scientific environment, about a reality or some, let's say, not so real situation with real risk of low intensity radiofrequency radiation for our health, and including these oxidative effects. So in 2015, we carried out meta-analysis on the issue. We actually tried to find all possible peer-reviewed publications which studied oxidative effects of low-intensity radiofrequency radiation. We have found exactly 100 experimental peer-reviewed papers. It's only about experimental papers. And actually, 93 of them demonstrated statistically significant oxidative effect, statistically significant change in these indices of oxidative stress, and only 7 actually didn't demonstrate. So it's kind of clear evidence that the situation is very, very typical for low intensity radio frequency or microwave exposure in living cells. By the way, in this meta-analysis, we have found both in vitro and in vivo studies. Most have done in animals, some have done in humans, and again, all of them were actually realized, were carried out under really low intensity radiation, much lower, typically much lower than safety limits of international guidelines. That is why we are clearly understand that these safety limits, it's not really safety according to their knowledge. Okay, and now just let's back to this idea that oxidative stress is connected with a huge amount of diseases and medical conditions. Keep in mind that low intensity radiofrequency radiation may stimulate, may produce oxidative stress. We are understand, we are in position of clearly understanding these all possible risks for actually our health, just due to this typical connection of oxidative stress conditions with other diseases and medical conditions. Some particular maybe unique situation is first of all about electrohypersensitivity. Again, you know that today more and more people actually suffer on these conditions when people have a kind of some allergic reaction on low intensity radio frequency or other types of electromagnetic radiations. But for my knowledge, mostly it's about radio frequency radiation. And again, 2 points. First of all, today, just very last years, we have evidence from some medical research that these people really have increased level of ROS. It may be just last year evidence about kind of real physical background of these conditions, yeah. And second, just indirect evidence that other allergic situation, for example, even from pollen, yeah, we have ROS to be involved in this allergic reaction. That is why it's very, very plausible that ROS have some, at least partial role in this development of electrohypersensitivity conditions. And next, definitely the most hot topic about potential carcinogenicity of radiofrequency radiation. You know, from 2011, the World Health Organization officially recognized, classified radiofrequency radiation as possible carcinogen to humans, but nevertheless, the discussion is continuing. And again, if we realize that low intensity radio frequency radiation can stimulate ROS overproduction in our cells, it's totally understandable because we have clear connection in many research and many analysis between ROS overproduction, actually oxidative stress, especially chronic oxidative stress, and risk of cancer development in our cells, in our organisms. I think, generally speaking, we may kind of conclude that, first of all, low intensity, so-called non-thermal radio frequency radiation is a real oxidative stressor for our cells in different ways, in few different ways. Yeah. Second, if we may have these oxidative stress conditions, especially during long-term situation, especially under chronic radiofrequency radiation exposure, we actually will get this chronic oxidative stress, which is critically harmful, critically risky, because it actually leads to depletion of our antioxidant system. And that is why we should realize that this low intensity radiofrequency radiation may be really, really harmful for our health, and just to keep in mind that we should be prepared, and we have a lot to do on this way. So I guess it's all for my presentation. Thank you for your attention, and let's hope that during the spring we will have much better situation, not only with radio frequency radiation, but with this pandemic situation. It's spring in Kyiv, in my city. So thank you very much. Goodbye.